selected or monitored, and how effectiveness or safety are defined, so that they can't really know what exactly is being studied. Pharma companies have traditionally argued that not only the trial patients, but the clinical trial data belong to them, that it's proprietary, even though the study's results impact millions. This is part of a kind of traditional secrecy in the field. Delaying protocol release conveniently means that it is a company's press releases, not the verified science, that dominate the public's all-important initial impression of its product. That the government regulatory agencies go along with all this, it is in fact standard practice, doesn't assuage the public. For many, it makes the whole process appear corrupt. And it doesn't help that, according to the conflict of interest disclosures of the authors of the Pfizer and Moderna vaccine clinical trials, some of the authors are employed by these companies and often have stock options. The essence of the scientific method is conducting experiments that everyone can objectively see and verify. Transparency is the bedrock of experimental science and the means to ultimately dispel doubt. Moreover, in terms of the scale of public involvement, the experience of the summer and fall of 2020 was unlike any other in the history of medicine. Never before had studies of this size and consequence been run so quickly, or a medicine been produced so quickly to be given to hundreds of millions of people. These studies were called Phase 3 clinical trials, and if they had positive results, then the vaccine could be given to hundreds of millions of people on the basis of an FDA emergency use authorization. But how long were the patients followed in these two studies after their second dose to assess safety and efficacy? 2 months. On that basis, the vaccines were given to over 100 million people. One must not confuse the perhaps immaterial fact that the vaccines were made quickly from the arguably more important fact that they were tested on people for only a short time. These vaccines were developed so quickly in part because the new mRNA technology allows quicker production, and because parts of the production lines that in the past were staged out over time were, in this case, set up simultaneously with the help of huge cash infusions. All else being equal, there's a serious argument that it might be hugely advantageous to be able to produce new vaccines so quickly. Quote, if you can intervene with, let's say, a 40% effective vaccine 4 months before you can intervene with an 80% effective vaccine, you save more lives with the 40% effective vaccine that's delivered 4 months earlier, close quote, Barney Graham of the National Institutes of Health pointed out. Quote, being fast in an outbreak setting in some ways is more important than being perfect, close quote. Still, it was obvious as early as the fall that some testing steps would be skipped. Quote, we'll have less safety testing than we typically would have, close quote, Gates noted. Quote, we just don't have the time, close quote. Must that be a problem? Why, especially during a pandemic, wouldn't we want to quickly distribute any vaccine that appears to work, even somewhat effectively, to those who are willing to take on any potential risks that may go with less safety testing? Some people might even decide for themselves that a raging pandemic is a dangerous enough threat to outweigh every other possible concern. But what we shouldn't do, if we want to maintain public trust and cohesion, is act as though there is no chance that any legitimate concern could ever possibly emerge, or that we know more than we really do after only 2 months of study. With complex biological systems, we simply can't presume that just because we have a fantastic idea for a treatment, the safety we hope for and see at the start will necessarily hold over time. Take the classic example of thalidomide. It was originally a sedative used for anxiety and later tried for nausea. It worked, leading some to theorize that it could prevent nausea in pregnant mothers. In practice, once on the market, it did. But it also caused serious birth defects in children. It took longer than 9 months and enough cases to realize that these side effects came from the drug, and even more time to overcome the drug company's opposition to the facts. The same applies to any of the major drugs pulled off the market for causing cancer, heart attacks, and diabetes. They don't always cause dire consequences immediately, or in everyone. Sometimes these drugs set a process in motion immediately, but it takes scientists a year or many years to pick up the trend in a population at large. Working from first scientific principles and based on what we already know, we can often develop a neat theory about what might work. But because we don't know what we don't know, it often doesn't turn out as we expect. That is why empirical science developed as a way to test our theories. Empirical science is always, by definition, science after the fact. This is especially important given the specific kind of vaccine that was being approved in the United States, the mRNA vaccine, which was a first of its kind. Instead of exposing a person to the virus itself in attenuated live form, like the MMR, or killed form, like the polio shot or flu shot, which is how many of the other vaccines we've come to know work. In the mRNA vaccine, a person is exposed to an artificially made genetic messenger, mRNA, that goes into their cells and directs them to make part of the virus, which then triggers antibodies. Early on in the rollout, both the pharma PR industry and the press emphasized how novel these vaccines were and how this unique technique would produce a vaccine so quickly. But when some side effects started to emerge and people got nervous, officials and the company's own PR teams changed their message. These techniques were now presented as not new at all, but as having been around a long time. The hesitance